We're going to learn about biological molecules today. I'm trying to revise for my exams. Best way to revise is to teach someone else. So I thought, let's make a video of it. We're going to start off with A-level biology, biological molecules. If you're doing AQA, it's 3.1. Let's get started! We need to know what a polymer is, because this whole section is about polymers. So a polymer is a big molecule made up of many repeating smaller monomers. Monomer is just like a small molecule, basically, like a amino acid, and then polymer is for example, a protein. You also need to know what a condensation reaction is and what a hydrolysis reaction is. So I'll go over that really quickly. A condensation reaction is putting two things together with the removal of water, and a hydrolysis reaction is breaking that bond with the addition of water. Now let's jump right into carbohydrates. Okay, what do carbohydrates contain? They contain hydrogen, oxygen, and carbon only, and they have the formula CH2O in brackets. That's just like the imperial formula if you do chemistry. So let's go on to those monomers, those monosaccharides. They have the general formula C6H12O6. You may have heard of that. That is because it is glucose and galactose and fructose. All of those ones I mentioned have the formula C6H12O6, even though it might be a 5-carbon chain like in fructose or a 6-carbon chain like in the glucose and the galactose. I need to know how to draw all of those. I'm going to put a picture of it up, and you need to memorize them. You need to know them for your exam if you're doing my exam. Disaccharides. This is made up from 2 monosaccharides formed with a what reaction? What did I mention before? A condensation reaction. And what is the bond in between those 2 monosaccharides? A glycosidic bond. Let's do some examples of disaccharides. So we've got maltose, sucrose, lactose. You need to know, what are they made of? Maltose is made of 2 alpha glucoses. Sucrose is made of an alpha glucose and a fructose. And lactose is made from a glucose and a galactose. And you also need to know how to draw those too. Anyway, you need to be able to tell the difference between reducing and non-reducing sugars. Now, to be honest, I don't really know what a reducing and non-reducing sugar is, but I don't think it matters. You just need to know how to test for them. So to test for a reducing sugar, you do the Benedict's test, where you add Benedict's reagent and you heat it, and if a red precipitate forms, you have yourself a reducing sugar. Now, if you do this to a non-reducing sugar, this won't happen. So you need to test it in a different way to test if you have a non-reducing sugar. So to do that, you need to add hydrochloric acid. This is to hydrolyze the bonds in it. And then you need to neutralize this acidic solution, because the Benedict's test doesn't work with acidic solutions. So you need to add sodium hydrogen carbonate. Then you need to add Benedict's reagent and heat again, and then a brick red precipitate may or may not form. If it forms, you've got yourself a non-reducing sugar. That's only after adding those 2 things. Then, lastly, in this section of carbohydrates, we have polysaccharides. So we have our starch, glycogen, and cellulose. So I need to know what these are made of. So cellulose is the only one that's found in plants, in cell walls, and that is made of beta-glucose. The other 2, starch and glycogen, are made of alpha-glucose. Both starch and glycogen are energy storage molecules. Starch is in plants, glycogen in humans. So they are both branch molecules because they need to store energy and be able to release energy quickly. So they're branched molecules and coiled together to be compact. Glycogen is more branched than starch, but this allows it to release energy faster. It has more ends, so the enzyme can get on it and release this energy. That is our carbohydrates. I hope this video helped you even a little bit. I'm sorry I'm bad at explaining things, but it kind of helped me to remember a little bit. And yeah, thanks for watching. If you're stuck or you need me to explain it again, put it in the comments, and maybe at the end of the next video, or I'll answer your comment below hopefully. And thanks for watching.